Hey science enthusiasts, welcome back to the LabMate channel where we're going to continue exploring the fascinating world of microbiology. Today we're going to dive into the universe of blood agar, so buckle up because we're about to embark on a thrilling journey. And I just want to apologize ahead of time. I do have a little bit of a cold, so you might hear me sniffle or even sneeze as I'm making this video. Alrighty, let's get to it. Okay guys, let's get started. So blood agar is an enriched medium that is used to culture fastidious bacteria, which are microorganisms that require an enriched nutritional environment to grow because of their complex nutritional environmental requirements. So the principle behind blood agar lies in the ability of the bacteria to interact with the blood's component, particularly the red blood cells within the medium. The medium's diagnostic value arises from the various hemolytic patterns displayed by different bacteria when grown on the medium. So the composition of blood agar consists of two main components, which is the agar base and blood. So usually nutrient agar or Columbia agar can be used. Nutrient agar contains basic nutrients that are essential for bacterial growth, while Columbia agar contains more complex nutrients and provides a richer medium for growth. Blood is usually sourced from sheep, goat, horse, or rabbits. So for preparation, first we have to prepare the nutrient or the Columbia agar according to the manufacturer instructions, or you can check out the previous video on nutrient agar and see how to prepare nutrient agar according to its different components. So once the agar is cooled to about 40 to 45 degrees Celsius after preparation, we have to add 5% sterile defibrinated blood aseptically and mix well. The agar is then poured into sterile petri dishes and under sterile conditions. Once the media solidifies, then it can be stored in a fridge before it is used. Very important part of preparing medium is that there has to be quality control checks that are done. So for instance, there has to be a control plate, which is an uninoculated plate that is incubated to check for sterility of the medium. This is by picking out a random petri dish. And then there also has to be a quality check. And this is done by choosing an organism such as E. coli, Staphylococcus aureus, and Streptococcus pneumoniae. That is what we usually use for blood agar. So what's next is basically the uses. What do we use blood agar for? So one of the important ones is for pathogen identification. So different hemolysis patterns aid in identifying pathogens and differentiating them from non-pathogenic bacteria, which is really important in the medical field. We can also use it to set up a camp test. We can also use it to grow up a wide range of pathogens such as Streptococcus pneumoniae, but it inhibits the growth of other bacteria such as Neisseria. Mueller-Hinton agar is the standardized method for antimicrobial susceptibility testing, but blood agar can be used for testing out fastidious organisms that may fail to grow on Mueller-Hinton agar. So some of the limitations of blood agar is that it is important to never use human blood in its preparation. Human blood is an inappropriate blood source and should not substitute the use of either sheep, horse, rabbit, or goat blood because human blood contains antibodies, antibiotics, and infectious agents that can present the results the wrong way. If I can explain, it's like it might end up inhibiting the growth of certain bacteria because you might have an antibiotic still present in the blood sample that you were using to prepare the medium. So it's better to use a standardized blood that is ordered from a reputable company / source. Another limitation is that it does inhibit the growth of certain bacteria such as Neisseria, because that there are inhibitors in the blood. These inhibitors can be deactivated by having a heated media of like blood agar heated up, which basically is chocolate agar, which will be discussed in a future video. And another thing to note is to never perform a catalase test directly on colonies, either directly on the blood agar plate or from colonies that come from blood agar plate. Instead, you're going to have to test, do a catalase test on samples from different type of, from a different plate. Because the blood agar, the blood contained in the agar will end up creating a false positive catalase test. Again, we'll go through that in a future video. So hemolysis. Hemolysis basically refers to the breakdown of red blood cells in the vicinity of bacterial colonies. This is very important when we're dealing with blood agar, because this is one of the important components or attributes of the agar itself, is that the ability for bacteria to hemolyze and hemolyze. So alpha hemolysis is the partial hemolysis, resulting in a greenish discoloration around colonies due to the conversion of hemoglobin to methemoglobin. As you can see in the little image that I created for you guys, is that there's this greenish kind of outline that is there. So this is kind of how it looks. You guys can refer back to the previous photo so you can see what it can look like on an actual sample. There's also beta hemolysis, which is a complete hemolysis, creating a clear or transparent zone around colonies due to the lysis of red blood cells. And then with gamma hemolysis, this is no hemolysis observed, no change in the medium around the colonies at all. So overall, blood agar is a vital tool in microbiology labs for bacterial cultivation and differentiation. It is unique composition and principle facilitate the identification of various bacteria based on hemolytic activity, and blood agar's versatility makes it an essential component of diagnostic and educational practices in microbiology. Check out the references for further reading. That is absolutely welcomed. Y'all can do that if you guys are interested in that. And stay tuned for more quick overviews of different agars. And don't forget to hit up the like button, subscribe to our channel, and share this video with your fellow medical and science enthusiasts. Don't forget to hit up the comment section or even communicate with me to tell me what kind of videos you guys want to see next. I will definitely try to post more consistently. It's just that life has gotten very hectic, very busy, and like I told you guys, I am not feeling super great. I do have a cold, and thank you so much for watching this video. Bye.